really, really interesting. So I talked about the oxidative phosphorylation that happens in the inner membrane of bacteria. And similarly, mitochondria perform the oxidative phosphorylation in case of eukaryotes. So they're called the powerhouse of the cell because they generate ATP molecules that are required for cell to function. So a single molecule of glucose produces about, or a single mole of glucose produces about 36 moles of ATP through glycolysis and Krebs cycle. You might have learned that in metabolism if you've taken a class on biochemistry or cellular physiology or metabolism. But I won't go too much into detail about that, but mitochondria is a site of oxidative phosphorylation. So it is a double-layered membrane or double bilayer membranous organelle. Again, that's an indication that it is performed, probably came from an ancestral prokaryote at some point in time. So the outer membrane is thought to be the original membrane of the eukaryote that engulfed the prokaryote. And then the inner membrane is the membrane of the prokaryote, the original. And then there is quite a bit of structural similarity to the membranes of mitochondria to bacteria. So there is the outer membrane, outer bilayer, and the inner bilayer is kind of folded into what are called cristae. So these folds are the location of all the integral membrane proteins that are required for oxidative phosphorylation and for the electron transport chain to occur. So it's got the lumen of the mitochondria contains its own DNA, its own circular DNA that's structurally or topographically similar to those from bacteria. And they also have a 70S ribosomes, which is again another similarity to bacteria. So yeah, the ribosomes and the mRNA that it requires to produce the proteins that are required for the mitochondria to function, and a circular DNA, which helps the mitochondria to replicate independently inside the cytoplasm. You have a question? Yes. Yes. Yeah, I believe so. I'm quite a bit sure. Yeah. No, there is. Mitochondria are fairly complex. They do a lot of other functions as well, not just cellular respiration. Like they produce some special type of fatty acid. There's definitely an exchange. It's definitely a symbiont inside the cell, meaning there is exchange of proteins, and so there are transporters in the membranes as well, and the inner and the outer membrane. Initially, it was thought that the outer membrane simply has pores, but then now they're discovering more transporters even on the outer membrane. So there is definitely export of molecules back and forth. It's not completely independent on its own. Enzymes, like transport of enzymes between mitochondria and the cytoplasm. Yeah, it's completely sustained, but I don't know specifically about enzymes. I don't know of any enzymes that are specifically being transported between back and forth. But it's definitely exchange of molecules. Yeah. Any other questions before I move ahead? Mitochondria are really, really exciting or really interesting, because if you see any of the older electron micrograph images of mitochondria, it's usually showed as like an ovoid or like a kidney-shaped organelle with lots of branching inside. While that is true, as we are discovering more and more information about mitochondria, we are realizing that they're highly dynamic organelles. Highly dynamic in the sense they move around inside the cytoplasm, and they don't look like those distinct oval-shaped organelles. They're in fact branching, and they divide and they fuse all the time inside the cytoplasm. And that characteristic is really important for a lot of different functions to keep the health of the cell. And a lot of mitochondrial defects or the loss of this ability to divide and fuse is been linked to a lot of different diseases in case of humans. For example, the Alzheimer's disease, cancer. There is a specific disease called a mitochondrial encephalopathy or whatever. Can't remember. I was just trying to quickly glance through. But there's a lot of different diseases that result from dysfunction of mitochondria. So mitochondria are not static organelles, or they don't look like single ovoid-shaped organelles as we are used to seeing. They're highly branched, highly dynamic. They constantly fuse, they constantly divide. And as you can see in that picture, it's from a Saccharomyces cerevisiae from yeast. The mitochondria is labeled with the green fluorescent protein, and you can see they're highly dynamic. They're constantly moving. There's a bud coming out of the mother cell, and then the mitochondria fusing and division is also important to make sure that the mitochondria or the healthy mitochondria get transferred to the daughter cell. And it also enables genetic complementation. I don't know if you've heard about the term genetic complementation. So it's a process where if one mitochondria is defected in one gene, and the mitochondria of the separate mitochondria in the same cell lacks another gene, the fusing of the two mitochondria allows the formation of a fully functioning fit mitochondria that can perform all the functions. And genetic complementation is like a normal kind of technique that's often done in genetics, where when two organisms lack a certain ability, you fuse the genes together so that the new or the hybrid organism has all the functions that are necessary. And mitochondria fusing is also known to enable formation of healthy mitochondria. And the experiment, the mitochondria mating assay, like this paper that I've mentioned, it's a really interesting paper. I highly recommend you read it when you have time, where they have shown evidence that mitochondria actually fuse together. So they had two haploid yeast cells, and then they each one of them was labeled with a different fluorescent protein. So one had red fluorescent protein, another one had green fluorescent protein, and the two haploid zygotes fused together to produce a diploid yeast cell, and then the fused mitochondria had a yellow signal coming out, resulting from the fusion of the mitochondria from the two different haploid cells. So it's a really important discovery, and if you have time, I recommend you read that. And yeah, I think, any questions about that?
I have a question.
Yeah.
Is the genetic complementation something that like scientists do, or did the cells do that by themselves?
Oh, the cells do that by themselves. And it happens naturally all the time. That's one of the reasons why I guess it happens in nature, because when they fuse together, you will come up with like the most fit mitochondria. It happens naturally. And scientists obviously do that as well, because we're constantly just simply learning from nature, all the tricks that nature already has. Does that answer your question?
Yes, thank you.
Anything else? Really interesting though, right? It's pretty neat. So let's move on to chloroplasts. So a lot of microbes that are eukaryotes carry chloroplasts, like for example, the photosynthetic algae, and cyanobacteria also carry, well, cyanobacteria are not eukaryotes, but then they also perform photosynthesis, but then because they're prokaryotes, they simply have the photosynthetic pigments and not chloroplasts per se, or the cyanobacteria themselves are like chloroplasts because, again, these chloroplasts originated from an endosymbiotic event where a eukaryotic cell that conducts oxidative phosphorylation simply engulfed a cyanobacteria that had the ability to photosynthesize. And the cyanobacteria simply decided to stay as a symbiont, offering the eukaryotic parent cell special abilities to become an autotroph, for example. So chloroplasts have a 3-membrane system. So they have an outer membrane and an inner membrane. Again, so the outer membrane coming from the original parent that engulfed the cyanobacterium, and the inner membrane is the membrane of the prokaryote. And then the third membrane system is coming from what are called thylakoids. So they are highly folded membrane system inside the chloroplast that carry photosynthetic pigments, like photosystems to carry out photosynthesis. And then again, so the outer membrane simply was believed to have pores for the exchange of metabolites, but then later scientists have discovered transporters even on the outside. And the inner membrane obviously has lots of different transporters necessary for the exchange of molecules between the cytoplasm and the inner lumen of mitochondria. And these thylakoids are highly dynamic. Again, they're constantly moving. They're not static structures because although we've seen pictures of chloroplasts showing stacks of coins, or they're called stroma, but then they are not static. They're constantly dynamic. They're constantly moving. And yeah, these membrane folds carry chlorophyll and they conduct photosynthetic reactions inside the cell. And again, there's another really important cool feature of chloroplasts is, as I mentioned, they came from a single endosymbiotic event where a eukaryotic cell simply engulfed a cyanobacterium. And then if you look at the structure of chloroplasts and structure of cyanobacterium, there's a lot of similarities. Some of them are kind of pointed out here. The cyanobacteria also have membrane folds inside that carries photosynthetic pigment. And then the cytoplasmic membrane are similar to in structure to those coming from cyanobacteria, or there's similarity in the membrane structure of chloroplast and cyanobacterium. And the chloroplasts obviously lost some of the peptidoglycan during evolution once they became symbiont inside a eukaryotic cell. But then the genetic material of chloroplasts have a lot of different peptidoglycan genes that are not entirely deleted, they're simply suppressed. They just don't get expressed. And the chloroplast also has its own genetic material, which is a circular DNA, similar to those in prokaryotes. And yeah, it's a lot of similarity, which again is a direct evidence for endosymbiotic theory. And another interesting thing I wanted to tell you is about this group of eukaryotes called Epicomplexa. I don't know if you heard about the phylum itself. It's a phylum of under protozoans that include organisms like Plasmodium falciparum, for example. Do you know what that causes? A really common tropical disease called malaria. You guys have heard about it? Yeah, so Plasmodium and then Cryptosporidium, a number of different pathogens come under this phylum of Epicomplexa. The reason why I'm mentioning is because it's really interesting. So Epicomplexa have certain organelles that are called epicoplasts. So epicoplasts are derivatives of chloroplasts, except they don't carry photosynthetic pigments anymore. They're basically derivatives of chloroplasts. I mean, we know protozoans, they don't perform, they're not autotrophs, they're pathogens, or they're simply present in aquatic systems. But apicoplasts are really neat organelles that are simply derived from chloroplasts, but lack any fluorescent or lack any chlorophyll or photosynthetic pigments. It is thought that apicoplasts are results of a secondary endosymbiotic event. So what happened was, it's believed that at the first endosymbiotic event, the eukaryotic cell basically simply engulfed the cyanobacterium, and then that eukaryote was engulfed by another one, where the chloroplast simply, the chlorophyll pigment was simply not necessary anymore. So they lost the ability to have photosynthesize, but instead, it just became an organelle that perform other functions necessary for the cell. For example, apicoplasts are known to have, known to help with causing infection in case of malaria. In case of Plasmodium, it helps in sugar metabolism and a number of other functions, but they lack chlorophyll. So yeah, again, if you want to read more about that, there's lots of information. Happy to provide resources, but I thought I'd mention it because it's really, really neat. Any questions? Nope. Okay, so let's talk about plasma membranes. So we talked about the plasma membrane in case of prokaryotes. We talked about the different type of transport mechanisms, and then the different type of transporters, and the fluid mosaic model, and how the membrane is always dynamic, consists of phospholipids and integral proteins that are kind of floating around that act as gates for passage of molecules to inside the cytoplasm. The eukaryotic plasma membrane is similar in the sense that it performs all the same functions as the plasma membrane of prokaryotes, except it performs certain additional functions. So functions like, I'll talk about that in a little bit, but it's a little bit more specialized. So it's got more proteins, because obviously eukaryotes are a lot more evolved compared to prokaryotes. So it and if you focus on the biochemistry of it, there are additional phospholipids. So for example, sterols like cholesterol and sphingolipids. So cholesterol is a type of sterol. It changes the membrane fluidity. And then sphingolipids are known to offer additional stability, because prokaryotes have a cell wall, and eukaryotes, a large number of eukaryotes don't have a cell wall, and they require additional stability. And in terms of the additional functions that eukaryotic cell membrane can perform are, so as I mentioned here, phagocytosis, pinocytosis, receptor-mediated endocytosis, and exocytosis. So we talked about the different type of transport mechanisms in prokaryotes, right? Could we mention some of those? What are the type of transport mechanism? They came under 3 specific categories. Does anyone want to mention? Let me think, like, do I remember them? So there was a simple diffusion, right, which does not require any energy. It's called passive transport. And then there was active transport, which requires energy. And then there was a third one. Bringing back any memory? Facilitated diffusion. Do you remember? There's specific transporters that kind of help move. So the molecule gets converted to another. So specifically, we talked about phosphotransferase system, where the sugar gets phosphorylated, then it's taken inside the cell. So eukaryotic membrane performs all those functions. And in addition, it performs phagocytosis, which is called the cell eating. You might have heard about this. This is like a really important phenomena that happens in our immune system where there are specific immune cells called phagocytes that engulf like a foreign material, like a bacteria, because these cells are huge. They can easily engulf a small bacterium and then digest them inside inside the cell, like inside the peroxisome or inside the lysosomes, or just get digested. So phagocytosis is called a cell eating. It's basically just engulfs the membrane, just moves around, just like how in the picture shown here, the organism simply becomes a vacuole, and then it essentially turns into a lysosome. It's a really interesting phenomena where all the digestive enzymes accumulate inside, the environment becomes acidic, and then the organism simply gets digested. And then a second type of membrane endocytosis is called pinocytosis. So phagocytosis is called cell eating because it's mostly solid material that gets engulfed, and pinocytosis is cell drinking. So usually if the cell needs to intake like sugars and amino acids and mostly nutrients that are dissolved or soluble nutrients, they get taken inside the cell pretty much in a similar fashion, except in this case, it would be a liquid that's taken inside. And then there's receptor-mediated endocytosis, where this again is important in immune function, where certain bacteria get attached to the immune cells via receptors. So our immune cells, like basophils and neutrophils, they have certain receptors on them that are able to specifically detect an infectious agent, like a bacteria, when it enters the cell. And then these receptors bind to the receptors on the bacteria, and then the membrane gets invaginated around that, and then the infectious particle is basically taken inside, and then it's later taken care of. So that's called the receptor-mediated endocytosis, because there are specific receptors that are involved in binding to the bacteria or whatever infectious particle. And then there's exocytosis, is simply the same way the cell takes in foreign material. It also has the ability to push out unwanted material. For example, waste material, usually toxic material. So bacteria have pumps that export toxic material. In case of eukaryotes, they use the process of exocytosis, where the molecule just gets closer to the membrane, and again, the membrane just kind of folds around the waste material and just gets pumped outside into the cell. All the toxic materials, unwanted waste, gets ejected by process of exocytosis. Any questions? Yeah. Oh, definitely. Yes. Yeah. Yeah. That's a good point. Anything else? Fairly straightforward. Cell wall. So cell wall, all eukaryotic cells, like animal cells, for example, don't have a cell wall. There's distinct classes of eukaryotes have cell walls. For example, algae have cell walls, diatoms have cell walls, and obviously land plants, higher plants have cell walls. So these cell walls are made of diverse materials in case of eukaryotes, not peptidoglycan. Some of them are like cellulose, for example, in case of fungi and plants, they're made of cellulose, which is a higher or complex polymer, sugar polymer. And then there is biogenic silica. In case of diatoms, the cell wall is made of silica. There's calcium carbonate. Agar, which we use as solidifying agent, is a gel-like material. Certain algae are made of agar. Usually marine algae have a lot of agar in them. That's where it's isolated from. And carrageenan is another gel-type material. We use it a lot in food industry as a solidifying agent. Most of the yogurts that you buy from the store contain carrageenan. Next time you read the components, you'll try to remember. So yeah, protists and algae mostly have carrageenan inside. And then chitin, yes, fungus, for fungi also have chitin. Some of the fungi have cellulose, some have chitin and glucans. So in case of eukaryotes, the cell wall is made of diverse type of material. So I just wanted to put a picture of a cell wall in case of brown algae, where it is made of alginate and sulfated fucan and fucalis. Don't get bogged down by too much details, but essentially the cell membrane is supported by a cell wall made of mostly, they're made of polysaccharides. As you can see, cellulose is a polysaccharide, and a lot of these carrageenans and agar, they're all polysaccharides, essentially polymerized with a different different kind of bonds or different arrangement of sugar molecules. So yeah, that's all I had about cell walls. Yeah, so I mean, I guess more than the cell wall, it's the extracellular matrix that that's kind of important, because the extracellular matrix, which is kind of pretty much loaded with all these gel type material, is what provides that stability for animal cells, because or eukaryotic cells, because eukaryotic cells don't have there's just a cell membrane which is highly fluidic and it's very dynamic. It doesn't really give a solid support. So which is why there is a extracellular matrix which is kind of continuous. So all the cells are present in this matrix. It's a continuous matrix. So cell wall is not really distinct, because in case of prokaryotes, just one, they're unicellular, they're distinct one cell. But prokaryotes, they can be multicellular. And so the cell wall is kind of, there is two cells, and the cell wall and the matrix is kind of in between all the cells. And usually, yeah, like I said, it's just a sticky mass of carbohydrate, which have different structures and different sugar molecules. So collagen is a great example. In case of humans, cosmetically, collagen has been given a lot of importance. It's essentially a protein that supports all the cells, or which holds the cells together, provides support for our skin. So skin has a lot of collagen. It's just a protein fibrous material which holds all the animal cells, or the cells together, the skin cells together. So fibronectins are interesting. So it's in the figure here. So the collagen fibers are, so they're present in the extracellular matrix, and then proteoglycans and collagens are attached to fibronectins. So fibronectins are important because certain bacteria attach to the extracellular matrix, because that's how they can get inside the cell, is by attaching to certain specific molecules. So human cells are rich in collagen, and this collagen is interconnected by fibronectin and proteoglycans. So certain bacteria make use of the specific receptors that bind to fibronectins, so that they eventually find their way inside the cell to cause an infection. Any questions related to that, or everything is pretty straightforward? So flagella and cilia. So certain protozoans, like Euglena is one example. So they are present in aquatic environments, and they swim. And flagella is a mediator, or a tool that allows the cells to swim. Similar to prokaryotic flagella, the motion of the flagella and how it propels the organism forward are all kind of similar, except there is a structural or biochemically they're different. So just like bacteria, that's a stiff whip-like structure composed. So in case of prokaryotes, it is made of microtubules. So we talked about the cytoskeleton. It's pretty much the same component that forms the flagella in case of eukaryotes. And it's got the 9+2 arrangement. So essentially there's 2 microtubule rods that present in pairs. And there are 9 of those different pairs arranged in a circular fashion. And at the center, there is another pair. So that produces, it's like a tube-like structure basically with like a central filament, which acts like a whip, which is like it's got a motor at the bottom and a basal body that sort of functions in a similar way as in case of bacteria. But in case of prokaryote, it's pretty much, even though it's biochemically has a slightly different structure, it helps the organism to swim, move forward. And then cilia are structurally identical, except they're much shorter. You might know that the epithelial cells that line our intestine they have cilia. So cilia, for flagella, there's just usually just one flagella or maybe a few, but cilia the cell is entirely covered by these short structures that helps with a number of different functions. So in case of intestine, they increase the surface area, and then they helped. So in case of respiratory tract is also covered with epithelial cells, which have cilia, and it helps in the movement of mucus, and if there's any sort of foreign particles, they kind of show like a sweeping motion almost. I kind of imagine that as you know how when the wind blows, the grass moves like a wave almost. That's how these cilia move, and they help in the movement of particles, any sort of dust or mucus. So that's mostly the function of cilia. And then the intestine helps in the absorption of food particles and motion, essentially just in general movement of the food in the gut. So yeah, they're flexible. They usually show like a waving motion, helps to move the food particle and mucus. And yeah, structurally they're identical to flagella, except they're much smaller and they're kind of specialized for a different type of slightly different function. They don't help with the swimming. Flagella helps with the swimming, and cilia is just for slightly altered function. Any questions regarding flagella and cilia? Yeah. Yeah, flagella, there are different type of flagella, right? I think you're talking about the amphitrichus or peritrichus probably, which covers the whole cell. And then cilia, you can compare them to the pili, probably. That's the closest, like the bristle-like structures. They would be similar to that, but they're not. They're performing completely different function. So pili helps with transformation and attachment and the competence system, which is it takes the DNA inside the cell. They also form the conjugation bridge. Completely different functions, and cilia is for completely different. But although they kind of look similar in the sense that it just completely covers the cell, but they're not the same. No, flagella are different. Flagella are much longer, and they help with the swimming. Yeah, the bristle-like structure. It's just the pili or fimbriae. They're kind of interchangeable. Or people say fimbriae are the bristle-like structure. The pili are the ones that help with the conjugation. Although structurally, there is not really that much difference. So the terms are interchangeable. I hope I'm not confusing you too much, but it's just the pili that makes the fuzzy structure that you're talking about. And flagella are 5 to 6 times longer than the size of the pili, and they help with the swimming. But pili helps with attachment, transformation, It also helps with the infection. Does that make sense?
Yeah, no, I think that helps sort it out.
Yeah, so they're totally different. Flagella and pili are completely different. Yeah, it's good.
Yeah, no, that helps. Thank you.
Any other question? Yeah. Sorry, say that again. No, they do have flagella. Cilia doesn't help with the motion. Yeah. 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 Ciliates would have a separate flagella for locomotion. Any other question? Is it time? Pretty close. I think that might be it for today. We're kind of coming close to eukaryotic cell structure and function. We'll start with a new unit in the next class. And Monday's lecture won't be synchronous. It won't be in person or on Zoom. I'll just upload a lecture. Okay, thank you. I will see you on Friday.
Hello.
Hello. Yeah.
Hi. Sorry, could you go back to the very first slide?
Oh, I'm sorry. What's
Could you go back to the very first slide about mitochondria?
very first slide of mitochondria? Sure.
Yes.
Yeah, that one.
Previous one, the one above that.
Oh, yes. Yeah. Sorry. There we go.
I might kind of miss that, but I thought we were talking about eukaryotic functions.
Eukaryotic. Yeah.
Right. So why would you say that they have 70S ribosomes, or did I kind of miss that? Well, I understand like how prokaryotic was engulfed by what is called.
Oh, yeah. No,
missed
you're
that,
right.
but-
So I hope I
but-
didn't confuse you. So the ribosomes that are specifically present inside the mitochondria are 70S. But
Okay.
the eukaryotic ribosomes, the main cytoplasmic ribosomes, are 80S.
Okay. Okay. Yeah, I was like, I've just kind of missed that. So thank you.
Yeah, no problem.
I have one question.
Sure.
I think you said, sorry, now I have to find it. Give me 1 second, sorry.
No, that's okay.
For the chloroplasts,
Mhm.
why does it not, like, why does it suppress the peptidoglycan? Because it's not going to make a cell wall.
It doesn't need a cell wall, right? So in case of bacteria, they need a cell wall because they need protection from the exterior because they're just organisms that are surviving outside on their own. For the chloroplast, once they're inside, they don't need any sort of protection. And the cell wall really doesn't have a purpose at that point, right? Because it's a symbiont. So there is the main parent cell that's kind of taking care of its. So yeah, I mean, all the functions of the cell wall is performing in of bacteria, they're pretty much irrelevant once the chloroplast is present inside the eukaryote. So those genes are suppressed and some of them are deleted. So those those genes don't even get expressed to produce the cell wall. Does that make sense?
Yeah, that's perfect.
Anything else?